Welcome to our very first lecture in microbiology. Before we get into Unit 1, Lecture 1, I'd like to familiarize you a little bit with our lecture schedule and how you know what we're doing each day in class. And this comes from your class calendar, which is posted on your learning management system. Now, if you click on a particular day of our class, for example, you'll notice that our first lecture in the first week of classes is Unit 1, Lecture 1. And when you click on that on your calendar, that'll bring you to this page, the lecture schedule for Unit 1. So in our first lecture, Unit 1, Lecture 1, we're going to introduce microbiology by looking at two SoftChalk lessons: basic groups of microbes, which includes an introduction to microorganisms, and the Human Microbiota and Microbiome, and then a second SoftChalk lesson on prokaryotic and eukaryotic cells. So on the day when that lecture is scheduled, it's your responsibility to go through these SoftChalk lessons, answer the objectives, learn and understand them. The next lecture in our first week classes says Unit 1, Lecture 2, so that day we'd be covering these three SoftChalk lessons: the three-domain system, bacterial sizes, shapes, arrangements, and forms, and the bacterial cytoplasmic membrane. The next lecture will cover these four SoftChalk lessons, the next lecture these SoftChalk lessons, etc. And notice that it takes us 5 lectures to complete Unit 1, which is actually our longest unit during our semester. Now, this table of contents you see here is designed for a traditional class that meets on campus. And during the first week on a traditional class, I do 2 of the SoftChalk lessons as flipped classes, which you see posted here in red. But of course, if we're doing this online as an online lecture, we're not doing flipped classes since you won't be coming to campus. You'll notice also that it says in red in many places, read on your own. These would be a SoftChalk lesson during a normal traditional class where I wouldn't cover that during my lecture, but it would expect you to read that on your own. These are usually pretty straightforward SoftChalk lessons, often involving some vocabulary and such. There is one important thing, though, in the red that you will need to keep aware of when we're doing the online lectures, and that's any item that says omit, like the very last one in Unit 1, Lecture 5. So anytime you do see omit in red after a SoftChalk lesson, you're not responsible for that lesson, and it won't appear on a lecture exam. So that's the way we know what we're doing each day in lecture, which SoftChalk lessons we're covering. And again, I can't stress enough how important it is to keep up with these every week. There's a lot of material covered in this class, and we really need to keep up with this. Once you start procrastinating or getting behind, you may not ever get caught up. So anyway, with that in mind, let's get into today's lecture, Unit 1, Lecture 1, Part 1, where we're going to be looking at basic groups of microbes, which also includes discussion on the Human Microbiota and Microbiome. And then Unit 1, Lecture 1, Part 2 will be on prokaryotic and eukaryotic cells. So let's get started on our first SoftChalk lesson, basic groups of microbes and an introduction to microbiology. Now, as I mentioned when I was giving my initial introduction to you and introducing the course, the first page of every SoftChalk lesson has bullet points and objectives. And these are the bullet points for our first SoftChalk lesson. Now, it's important that you start out reading these because these give you an indication of what we're going to be doing and covering in that particular SoftChalk lesson. So important key bullet points here in our first SoftChalk lesson is that microorganisms are typically too small to be seen with the naked eye, which is why they're called microorganisms, micro referring to small. We're going to learn that there are 5 major groups of microbes, namely bacteria, fungi, viruses, protozoa, and algae. So we'll be covering that shortly. We're going to learn that the vast majority of microorganisms are not harmful, but rather beneficial. we're going to learn about the microbiota and microbiome. Microbiota refers to all the microorganisms that live in a particular environment. For example, the human microbiota are the microorganisms that live in and on our body, whereas microbiome refers to the entire collection of genes found in all the microbes that are associated with a particular host. We're going to be looking in some detail at the microbiome of the human body, especially that of the gastrointestinal tract. The microbiome play critical roles in digestion of many foods. They regulate many of our own metabolic pathways, as we'll see, and they play an important role in regulating our body's immune defenses. And then a few definitions we'll need to be familiar with. Symbiosis is living together of two dissimilar organisms, such as a human and a particular bacterium. Mutualism is a symbiotic interaction between two species that's mutually beneficial, where both organisms benefit from living together. Whereas commensalism is a symbiotic interaction between two different species, where one organism lives on or within another organism, and that organism drives benefit, but it doesn't harm or benefit the host necessarily. So those are the key bullet points that give you an idea of what we're going to be covering in this very first SoftChalk lesson. Now, also, as I mentioned in introducing the course and in your Getting Started course tour on how to prepare for lecture, what you're responsible for in each SoftChalk lessons are the detailed learning objectives for that lesson. So once you read your bullet points, you want to take a close look at these objectives, because this tells you what you'll need to know for the exam for that particular SoftChalk lesson. And many people do this by writing them out first and then going through the SoftChalk lesson and answering them. That way you're getting the benefit of writing out the answers to help you learn vocabulary and how to spell words. You're reinforcing it, because the very worst way to learn is to try to read something over and over again, which is not very effective. And then you want to keep going over these objectives throughout the whole unit, reinforcing them and learning them so that you can answer them on the exam. So notice the detailed learning objectives for this first SoftChalk lesson. Says you should be able to state the 3 harmful effects and 4 beneficial effects associated with the activities of microorganisms. You should be able to define microbiota and microbiome. You should be able to define symbiosis, mutualism, and commensalism. You should briefly describe 2 different beneficial things the human microbiome does for the normal function of our body. State several diseases that are associated with a change in our normal microbiota. And finally, list and recognize a description of each of the 5 basic groups of microorganisms. So those are the things we're going to be learning in this first SoftChalk lesson. Now, before we get started on the first SoftChalk lesson and the content, here's a little think, pair, share question, which we'd normally be doing in class. But of course, I'll just mention this to you online since this is your online lecture. So this picture here shows you a tube of Escherichia coli, a bacterium we would normally work with quite a bit during the semester. And this particular tube has 8 milliliters of E. coli in it. Now, a milliliter is about 10 drops, so there's about 80 drops of bacteria in this tube. And so the question is, are the number of bacteria in this tube equal to the number of people in Baltimore City, the number of people in Maryland, the number of people in North America, or the number of people in the world? And normally we would go through and ask for a show of hands on that. But the correct answer, in fact, is D, the number of people in the world. Because 1 milliliter in this tube, which is about 10 drops, contains roughly 1 billion bacteria. So this tube contains 8 billion bacteria. And the latest estimation I saw of the world population is about 7.8 billion. So this tube contains more bacteria in it than the entire population of the world. And that gives you an idea of how abundant these microorganisms are and how they become the dominant life form on Earth. We just don't realize it because we don't see them most of the time since they're microscopic. So if you accidentally were to spill that tube of E. coli in the lab, you'd be spilling a number of bacteria equal to roughly the population of the world. Now that we've looked at the bullet points and the detailed learning objectives for our first SoftChalk lesson on the basic groups of microbes, let's get into the actual content. So our first topic in our first SoftChalk lesson of Unit 1, Lecture 1, deals with the 5 basic groups of microorganisms that we'll be talking about during our class. So our first group are the bacteria. And bacteria are typically unicellular or single-celled. They are microscopic almost always. They are prokaryotic organisms. And in fact, our second part of Unit 1, Lecture 1, will compare pro and eukaryotic organisms, although you should be a little bit familiar with that since that was a topic you had in your introductory biology course that was a prerequisite course for microbiology. So they're single-celled microscopic prokaryotic organisms, and they replicate by binary fission, which we'll talk about later on, which means each cell splits in 2 during their asexual reproduction. And we see an example here of a bacterium. This is Staphylococcus aureus that we'll be looking at quite a bit during lab, and we'll have one lab dealing with the Staphylococci. And we see them as they would appear under the microscope when you'll see them in lab. In this case, a gram stain of Staphylococcus aureus. We'll learn a little later on that cocci are spherical bacteria, and staphyl refers to a bunch of grapes. So these are bacteria that typically appear in little grape-like clusters. So this is the way they would appear magnified under the microscope 1,000 times. They're magnified even more on the computer screen here. And this is a scanning electron micrograph of Staphylococcus aureus where we can see a much higher magnification. Again, we see the round shape or coccus shape in the grape-like bundles. So it gives you an idea of what a typical bacterium looks like Staphylococcus aureus. A second group of microbes we'll be studying are certain fungi, specifically the yeasts and the molds. We're not going to get into the larger fungi like mushrooms and things like that, but there are yeast and molds that are involved in infectious diseases as well as in a number of beneficial products. So starting with yeasts, which we'll have in one of our labs. Yeast are typically single-celled or unicellular microscopic eukaryotic fungi, and they reproduce asexually by a process called budding. And this is where, following mitosis, a bud forms around one of the nuclei, pinches off, and becomes a new yeast. For example, in figure 3 here, we see Saccharomyces cerevisiae, or baker's yeast, as it would appear stained under the microscope. We see how much larger the eukaryotic yeast is than these tiny prokaryotic bacteria. One of the things we'll actually be stressing in lab 1 in our lab manual. And we can see buds forming involved in reproduction. And then this is another yeast, Candida tropicalis. Again, you see the oval budding yeast cell. And we'll also learn that yeasts will often produce filament-like structures called pseudohyphae and then hyphae, where they elongate and produce filament-like structures. Molds, on the other hand, are what we call filamentous fungi. And all fungi, of course, are composed of eukaryotic cells. So they produce long thread-like structures or filaments, and they reproduce by producing very large quantities of asexual reproductive spores. And one of our labs will deal with molds. For example, this is the Penicillium mold. And here we see how it looks under a conventional light microscope. And we can see the spores, in this case called conidia spores, that it produces for reproduction. And then here's the same mold under a scanning electron microscope, magnified a great deal more, where you can see the asexual reproductive spores that are being produced by the mold. Another group of microbes we'll be looking at during the semester are viruses. Now, viruses are typically submicroscopic, and that means we can't see them with a conventional light microscope. They're too small, although we can see them with an electron microscope. And another very important thing about viruses, as we'll be learning when we get to pro and eukaryotic cells, is that they are acellular. They're not composed of cells. So we call them acellular infectious particles. And they can only replicate inside a living host cell. We'll learn why later on, but since they aren't cells, they have no metabolic machinery, no cytoplasm, no way to make energy, no way to make cellular parts. So they have to get in our cell and convert our cells into factories for making virus parts and putting those parts together. And the vast majority of viruses have either DNA or RNA, but not both. Here we see a couple electron micrographs. Figure 7 is an adenovirus. Adenoviruses, there's a number of those that cause respiratory infections. There's one that causes pink eye. Some cause GI infections. And over here we have HIV. And here's the human immunodeficiency virus, and it's in the process of infecting a T4 lymphocyte, as seen under a transmission electron microscope. Another group of microbes are the protozoa. And these are typically unicellular microscopic eukaryotic organisms, but unlike fungi, they lack a cell wall. And we'll be discussing these in one of our labs as well. So for example, here's an amoeba, which some of you have probably seen in a biology class, and a paramecium. Those are both protozoa. And then our final group of microbes are algae. Now after today, we won't be talking about algae because we're really zeroing in on microorganisms involved in infectious diseases. And of course, algae, being organisms that carry out photosynthesis, are not very good at being pathogens. But algae are microorganisms, many of them are. And these are eukaryotic microorganisms, but they carry out photosynthesis. And here we see under a conventional light microscope a couple of algae in figure 11, Spirogyra. And you can see the green chloroplasts used for photosynthesis that are arranged in spiral filaments inside. And then a single-celled alga called Euglena, where you can see the chloroplasts inside involved in photosynthesis. Now, one of the things we really want to stress during the course is just how small these microbes are. The TPS question I showed you a little earlier mentioned that that tube of E. coli with 8 milliliters, or about 80 drops of E. coli, contained roughly 8 billion bacteria, kind of equivalent to the population of the world. And they're so tiny that that many can fit in a very small space. And there's a couple of nice little videos that illustrate this. Let's take a look at this first one here. So using a little zoom vision here, on this scale, this would represent a coffee bean, a grain of rice, and a sesame seed, to give you an idea. And what we're going to do is start zooming in and looking at things smaller than that. So now we're down to a grain of salt compared to a sesame seed. And now we see one of the largest amoeba called Amoeba Proteus. So you see that it's a little smaller than a grain of salt, and that's a pretty big protozoan. There's a paramecium and a human egg for size comparison compared to a grain of salt. If we magnify some more, now we can see a skin cell and a sperm. Keep magnifying. Now we can see a red blood cell and some baker's yeast, what we saw in one of the pictures earlier. And now we're getting down to our first prokaryotic microbes. Here's E. coli, a bacterium we'll be working with quite a bit during the semester. And notice that mitochondria are about the size of an E. coli, because it's thought that mitochondria actually evolved from bacteria. We keep zooming. And now compared to the bacterium, now keep in mind that bacterium, if we back it up a little bit, get this saturated. There's a skin cell right there, and right there's the E. coli. So a bacterium typically looks like a little dot or a little dash on a human cell. That's how small the prokaryotic bacteria are compared to a eukaryotic skin cell. And now we start getting into some viruses. There's the Measles virus compared to E. coli. The Influenza virus, HIV, and this is a virus that infects bacteria called a coliphage, and this happens to be a virus that infects E. coli. So notice just like E. coli looks kind of like a dot on a human cell, a virus looks like a small dot basically on a bacterium in terms of size comparison. Then we get into some smaller viruses like the hepatitis virus and the rhinoviruses that cause a lot of the common colds. And notice these are about the size of a human ribosome. And then we have an antibody molecule, some transfer RNA, a molecule of hemoglobin, some phospholipids, a glucose molecule, and all the way down to a carbon atom. So again, we get an idea of just how small these microbes are compared to things we can see with the naked eye. There's a nice little illustration here that shows viruses pretty nicely. Let this load in. So it starts out with what we saw in the previous one. This is a rhinovirus. There's about 100 different rhinoviruses that cause the common cold, and about 100 other viruses besides rhinoviruses that cause the common cold. But that's a pretty small virus. This is only 0.03 micrometers. Now a single Staphylococcus is about 1 micrometer. Then there's a poliovirus, about the same size as a rhinovirus. Here we have the influenza virus, quite a bit bigger than the poliovirus and rhinovirus. The rabies virus. And here's the bacteriophage that infects E. coli. So it's actually a pretty big virus compared to a lot of human viruses, but much smaller than the bacteria, as we'll see coming up. Smallpox virus. And now finally we see a bacterium, a single Staphylococcus, which is about 1 micrometer in diameter. So again, we see how small these viruses are compared to a fairly small bacterium, a 1 micrometer in diameter coccus. Lactobacillus. Here's E. coli that's typically about 2 micrometers long. So there's the virus back here that infects E. coli. So there's 3 bacteria compared to a red blood cell, compared to Baker's yeast, a eukaryotic fungus, compared to a skin cell and a sperm. And we'll stop it there, but that again gives you a nice idea of just how small viruses are compared to bacteria, compared to eukaryotic microbes, compared to human cells. And remember, as you go through here, there's self-checks along the way, so you should be clicking on those and trying to answer the questions, and try to answer them after studying it, but don't just keep clicking on it until you find the right answer. you want to use this as kind of a self-quiz. You're not graded on these, but if you understand that section on the 5 groups of microorganisms, you should be able to do well in that little self-quiz. And if not, go back and repeat the lesson, learn it some more, try the quiz again. Now, of course, since most of the people taking microbiology here are going into some allied health profession, we're especially concerned with looking at the fundamental concepts of microbiology with regard to how they relate to human health. So what we're going to try to do is kind of the big picture of this course is look at the better understanding of the total picture of infectious disease, what we call host infectious agent interactions. And in fact, this little box here shows you what's on the last page of your syllabus, what we call the big picture of infectious disease and kind of my approach to the course. So let's just review that briefly, although that is in your syllabus. So my approach again is kind of the big picture of infectious disease. Try to give you all the basic fundamental concepts you would get in any college level microbiology course, but make it relevant to the population of students taking this course, and that's the process of infectious diseases. So there are several parts to this big picture. We're going to be looking at the microbe side of the story. And what we're looking at there is trying to understand why only some microbes are potentially harmful to humans, whereas the vast majority aren't. What makes them different? So what can they do to colonize our body and cause harm? And then we're going to be, and units 1 through 4 will take care of that. Then we're going to look at the body side of the story, ways in which our body defends itself through our immune defenses and such. And units 5 and 6 will cover that. Throughout the course, we're going to look at ways we can artificially help the body defend itself, either by removing the microbes or by enhancing our body defenses. And also, and especially in today's lecture, we're going to look at the relationship between the human microbiome and human health. So those are kind of the 4 parts of the big picture that kind of interlock together to determine whether or not a person gets an infectious disease. Excuse me. So in units 1 through 4, we're going to look at the microbe side of the story, why certain microbes have potentially more ability to be harmful than others, because the overwhelming majority of microbes are harmless to humans. And of course, they're critical to life on Earth, doing things like recycling nutrients and such, as we'll see a little later on in our SoftChalk lesson here. So we're going to learn about the major groups of microbes, what they're composed of chemically and structurally, how they carry out their metabolism, how they reproduce. And then we're going to look at the various factors that some microbes possess that enable them to cause disease. And we'll also learn how through mutation and genetic recombination and natural selections, they can adapt to resist our control attempts. Then the second part of the course, which we cover in units 5 and 6, are the body side of the story, ways in which the body is able to defend itself naturally against infectious disease agents. And we have phenomenal body defenses in our body. And not only do we use these to get rid of infectious disease agents, but to get rid of our own harmful cells, things like cancer cells and infected cells. So we do this through 2 immune systems, the innate immune system, which we'll take up in unit 5, and the adaptive immune system, which we'll take up in unit 6. And we'll learn later on that the innate immune defenses are the ones we're born with. They're going to include things like anatomical barriers to infection, the ability to mechanically remove microbes from the body. We're going to learn about how they stimulate an inflammatory response to help get rid of the microorganisms, things like phagocytosis, the complement pathways, fever, all parts of our innate immune system. And then in Unit 6, we'll look at the adaptive immune defenses, such as antibody production or humoral immunity and cell-mediated immunity, where we have a cellular response. And then throughout, we're going to look at ways in which we can artificially help the body defend itself by removing microbes or enhancing body defenses to avoid or reduce the risk of infection. So, of course, there are agents we can use like antiseptics, disinfectants, physical agents we can use to control microbes like heat and cold, antimicrobial chemotherapeutic chemicals and antibiotics we can use. And also we're going to be looking at ways we can currently or maybe in the future improve or restore the body's immune responses through techniques like immunization, adaptive immunotherapy, immune modulation, and such. And finally, the relationship between the human microbiome and human health. And we're going to be covering that actually in this current SoftChalk lesson in quite a bit of detail. But we'll learn how critical the human microbiota and microbiome are to our good health. So those are the parts we're kind of putting together here to make up this course so we can look at all the general concepts and principles that would be in a college level micro course, but try to make it relevant to the majority of our population. And of course, in the lay public, a lot of people think of microbes as harmful. We always heard them referred to as germs, and we only hear about the microbes when they're harming the body, because of course they can cause infectious disease in humans. They can cause infectious diseases in other animals. They can cause a lot of agricultural loss, infectious agents that infect plants. And of course, since their main job in nature is to recycle nutrients, they cause a lot of spoilage of food unless we take precautions to prevent the bacteria from growing on the food, such as canning or refrigeration and such. But of course, what we're going to be stressing here is that the vast majority of microorganisms are not harmful, but rather many of them are beneficial. And in fact, without microbes, there would be no life on Earth. They influence pretty much every activity going on in Earth. So as kind of a brief introduction before we get into the microbiota, let's look at some of the benefits associated with microbial activity on our planet. First of all, of course, food production. There's a lot of food products that employ microorganisms in their production. Microbial fermentation processes are used to produce things like yogurt, buttermilk, cheeses, our alcoholic beverages, leavened bread, things like sauerkraut, pickles, kimchi. These are all fermented food products. Microorganisms can be used in energy production and cleaning up the environment. Methane gas or natural gas is a product of methanogenic microorganisms. Many of the aquatic microbes, especially the algae, capture light and store it as molecules that's used by food by other organisms. Things like animal waste, domestic refuse, biomass, and grain can be converted into biofuels like ethanol or methane. And through a process called bioremediation, we can use microorganisms to remove or neutralize pollutants from sites that have become contaminated. Microorganisms are also necessary to sustain agriculture. It's the microorganisms that recycle nutrients in nature. Remember, in biology, you learn that all things are recycled except energy. Energy, of course, can only be converted from one form to another, but everything else is recycled. When living organisms die, they're recycled by microorganisms to recycle the nitrogen, the carbon, the sulfur, and all the other nutrients found in living organisms. And these can be used then by plants for their growth, and plants can be used to feed animals. So they are important in agriculture. And of course, there's a lot of useful gene products or products that can be used, produced through bioengineering, making specific enzymes, making antibiotics, making vaccines, making medications like human insulin, human interferons, human growth hormones. So those are our 5 basic groups of microorganisms, an idea about how small they are, and some of the beneficial things that these microorganisms do for us in nature. Our second topic in our first SoftChalk lesson of Unit 1, Lecture 1 deals with the Human Microbiota and Microbiome. Now this is our chance actually to talk about all the good things that microorganisms do, especially for our body. Because of the nature of this course, since most of our students are going into nursing or becoming PAs or going to dental hygiene school or some other allied health profession, we tend to stress the harmful effects of microbes and the microorganisms that causes infectious disease. But again, the vast majority of microbes are not able to cause infectious disease. So this gives us a chance to look at some of the good things that microorganisms do and how critical they are to the functioning and well-being of our body. Or as we say here, where would we be without microorganisms? Now, first, some ideas as to numbers. It's estimated that the typical human body has about 30 trillion human cells, and the vast majority of those are red blood cells. But our body also contains approximately 39 trillion bacteria and other microbes. So as you see, we have slightly more microorganisms in our body than we have human cells. And during a good part of our day, we are in fact more prokaryotic than eukaryotic if we look at simply the number of cells that make up our body and our microbiota. So a couple of terms, these are terms that were in your detailed learning objectives. Microbiota is used to describe microorganisms that typically inhabit a particular environment. Could be the body, or maybe inhabit soil or a body of water. Of course, what we're going to be looking at is the human microbiome, those that inhabit our body. Now, sometimes these are also referred to as the microbiome. Ecologists tend to use microbiome to describe the organisms as well that inhabit a particular environment. But in microbiology, we tend to use microbiota to refer to the microbes that are inhabiting an environment. And we use the term microbiome, which we'll talk about down here a little further, to describe the collective genes associated with the microbiota. So for our purposes, microbiota will describe the microorganisms that inhabit a particular environment, and in this case, the human body. Now, of course, this varies greatly from person to person. It varies with age, height, weight, individual factors, but it's estimated, in fact, that if we took all the microorganisms in our body and collected them, they would weigh about 2.5 pounds. Now, we consider that a bacterium weighs about 1/trillionth of a gram. That is an awful lot of microorganisms, like we said, about 39 trillion bacteria and other microbes. Now, just to give you an idea of various parts of the body, if we look at the gastrointestinal tract, let's start with the third bullet point here. The upper small intestines, the duodenum and jejunum, these are actually fairly sparse in microorganisms. There's between 1,000 and 10,000 bacteria per milliliter in the small intestines. And although 10,000 seems like a lot, there's not really very much compared to many parts of the body. When you get to the lower small intestines, the ileum, it rises substantially. Here we have about 100 million bacteria per milliliter. But the area of the body that really teems with microorganisms and constitutes the majority of the microbiota in our body is actually the colon. The colon has approximately 100 billion bacteria per milliliter. We think of that in terms of feces. A milliliter would weigh about a gram. A gram is about the weight of a paperclip. And that, so some feces that weighs about what a paperclip would weigh would have about 100 billion bacteria. And so that's where we find the vast majority of these 39 trillion bacteria. And that's why we are mostly prokaryotic, more prokaryotic than eukaryotic anyway during a good part of the day, but a lot of that depends on your biological functions. So for example, if you're sitting on the porcelain throne and make a deposit, you get rid of an awful lot of bacteria. For a short period of time after that, you are now more eukaryotic cells than prokaryotic cells. But within a matter of hours, that all those bacteria have regrown and have replenished the colon. Other parts of the body, dental plaque has an estimated 10 billion bacteria per milliliter. Saliva has an estimated 1 billion bacteria per milliliter. And again, a milliliter is about 10 drops. The skin has about 10 billion bacteria per square meter of skin, with an average adult having about 1.8 square meters of skin. So although 10 billion per square meter seems like a lot, it's actually fairly sparse compared to places like the colon. But anyway, as you see, our body is covered with and filled with microorganisms, primarily bacteria. And there's a symbiotic relationship between our body, the host, and its microbiota. And this is one of our definitions we need to know. So symbiosis is where two dissimilar species or organisms live together, like a particular bacterium living with a human. And this can be one of two possibilities. If the symbiosis is mutualistic, what we call mutualism, mutualism is where the two organisms live together and both benefit mutually. So both of the organisms are mutually benefited by the association of living together. Now, in other cases, that symbiosis is commensalism or commensalistic. And that's where one organism lives on or within another and derives benefit, but it doesn't necessarily harm nor benefit the host. So a lot of the bacteria living in our body are mutualistic, where both the body and the bacterium are mutually benefiting one another. But there's a lot of other microbiota that are commensalistic, where the microorganisms are benefiting, but they're not harming nor are they benefiting the host. But we now know that this complex mutually beneficial symbiotic relationships between humans and our natural microbes is critical to our good health. Now keep in mind we have roughly, as we set up here, 39 trillion bacteria in our body, and each one of those has many genes. A typical bacterium has typically around 3,000 genes. So there's millions of genes associated with the microorganisms living in our body, that is our microbiota, especially in the intestinal tract where we have the most dense population. And so the gene products of the microbiota actually aid in the digestion of many foods. They regulate a lot of our metabolic pathways, and they also regulate our immune defenses. So the collective genes of the microbiota we call the human microbiome. So again, we're using microbiota to refer to the microorganisms that inhabit our body, and their collective genes and gene products are the microbiome. Now, it's thought that the human genome has between 20,000 and 23,000 genes, but it's estimated that there are 5 million to 10 million genes from over 1,000 species of microorganisms that constitute our human microbiome. So there's about 300 non-human genes in the human body for every human gene. And these microbial gene products play critical roles in the normal function of our body. So one thing we know that the microbiota and their gene products do is that they regulate a lot of our metabolism. So again, this is essential to human health. And this is done many ways. First of all, the digestion of many foods, especially polysaccharides that come from plants. There's hundreds and hundreds of polysaccharides in the plants we eat. And the human body only has something like 12 genes that can break down carbohydrates. So we could break down very few plant polysaccharides. if we didn't have microorganisms. It's primarily the gene products, the enzymes of our microbiota that are breaking down plant polysaccharides into useful metabolites that we can use. Also, the microbiota and the microbiome regulate many of our own metabolic pathways. So the metabolism of many of the substrates we use during enzyme reactions are carried out by a combination of genes from both the microbiome and the human genome. It's both the human gene products and the microbial gene products working together to regulate our metabolism. This is especially true in the intestinal tract, where there's a constant communication between the microbial species themselves. We'll learn later on under quorum sensing that they talk to one another chemically, and that influences their activity. But there's also communication between microbial cells and human cells. So there's a lot of things that influence our biochemistry and our body's susceptibility to disease. And there are multiple factors that can change our microbiota. They include things like diet, antibiotic use, disease, lifestyle, even the person's environment. All of this can alter the composition of our microbiota within the GI tract and therefore influence our biochemistry and our body's susceptibility to disease. And there are many metabolic disorders that are associated to actually with alterations in our microbiota. These are a lot of these lifestyle diseases that many of us now have because of the nature of our lifestyle. Metabolic disorder like diabetes, non-alcoholic fatty liver disease, hypertension, obesity, gastric ulcers, colon cancer, even mood and behavioral changes through hormone signaling have been linked to alterations in our microbiota. And besides regulating our metabolism, we also know that the microbiota is critical to regulating our immunity. So we know that many of the bacteria that live in and on our body, along often with parasitic helminths earlier on, co-evolved with the human body over about 200,000 years. So that the human microbiota now plays a significant role in regulating our immune responses. And what the microbiota do is they provide a nice series of checks and balances. Now we have to be careful. Our immune system is phenomenal, but if our immune system gets too aggressive, it can attack our own cells, causing autoimmune disease and destroy the body's own cells. So one of the things the microbes have done is to help regulate our immunity so that we don't become too aggressive. And that was actually to their own benefit because we don't want to kill our good microbiota, the ones we need for normal function. So the body co-evolved so that we wouldn't attack the good bacteria that we need. And in so doing, that also dampened the immune responses so that we also have less chance of attacking our own cells. But at the same time, our immune system has to be aggressive enough that it can recognize and remove harmful pathogens. So we have this nice balance going on in our body where the microbiota and the human cells influence one another so that we don't get rid of the good bacteria we need generally, but we do have a strong enough immune response to attack harmful pathogens, but not so strong that it can attack our own body by mistake. So we know for sure that the microbiota affects the development of our immune system, and also our immune system influences the composition of our microbiota. And of course, if we considered the first humans that were walking the earth, starting out as hunter-gatherers, and then eventually developing into a more agrarian society, and then into a more industrialized society, these big changes in humans over the eons have affected our microbiota by affecting our ability to colonize and maintain proper organisms. So when the body And the microbes co-evolved over 200,000 years. During most of those 200,000 years, humans were hunter-gatherers or had a more agrarian-type lifestyle. And of course, as humans changed their environment and their living styles, that then affects our development of microbiota. So the exposure and colonization with these once common human organisms that everyone had through most of human history has drastically changed over a time through a lot of factors. Simple things like less exposure to mud, animal, and human feces. Of course, in a hunter-gathering or an agricultural type society, people were exposed to mud and dirt all the time, and of course exposed to animal feces, and often, since we didn't have the sanitation we do today, to human feces. But that means now, as that changed, of course, today we have much less exposure to dirt and mud, since the majority of the country lives in cities where we have concrete and roads and all of that. We don't have the traditional animals that humans were around through a good part of human history. It's more pets. And of course, with good sanitation, we have less exposure to human feces. Now, of course, with that good sanitation and less exposure to human and animal feces, that has prevented a lot of gastrointestinal infections that traditionally infected humans through the fecal-oral route. But also by having less exposure to things like mud, animal feces, and human feces, we get less exposure to the bacteria, the majority of the bacteria in feces that's actually beneficial bacteria. And of course, we couple that with increasing antibiotic use. The antibiotics can't tell the good bacteria from the bad bacteria, so they are equally good at destroying normal microbiota. Improved sanitation, like I mentioned. That has helped in a great deal in terms of intestinal infections, but also there's less exposure to all the good microorganisms that would be found when sanitation wasn't what it is today. A big player is change in the human diet. During most of those 200,000 years, of course, the diet is quite different from modern humans' diet. Our modern diet today, of course, is largely based on fat, sugar, salt, and things like that. These are not things that humans normally ate during most of their history. And so the microbes that co-evolved with the body co-evolved on the human diet of that time, or is basically plant-based with a little bit of meat, not all the sugar, the salt, not all the fat, and all the things that are part of our diet today. As our diet changes, that changes the food source in our intestines. And so now other bacteria can grow better on those fatty foods or with all the excess sugar and such. And that changes the ability of bacteria to compete. Now we're going to learn later on, when we get to pathogenesis of bacteria, that most of our normal good microbiota are anti-inflammatory bacteria. They're bacteria that don't stimulate an inflammatory response. Our body's immune system evolved so that we don't recognize the microorganisms and then stimulate an inflammatory response to bring defense cells to get rid of them. So we consider these as anti-inflammatory bacteria. But the bacteria that often now live in our intestinal tract, especially in the colon again, are ones that have evolved to subsist well on the foods that we are currently eating. And so our body hasn't co-evolved to accept these bacteria, and it recognizes them as foreign and stimulates an inflammatory response. So a lot of these lifestyle diseases we see in modern society are due, in fact, to constant inflammation and the release of free radicals as part of our immune system that were originally designed to get rid of harmful bacteria. But now we're producing these all the time because a lot of our microbiota is no longer non-inflammatory, but rather inflammation-inducing bacteria. Another factor, increased rate of cesarean sections, decreased rate of breastfeeding, improved methods of processing and preserving foods. Again, that has allowed foods to last longer and prevented pathogens that might be associated with foods from developing, but also we aren't being exposed to all the microorganisms that normally would be growing on raw foods. So what we do know is that with all these changes in our lifestyle in the developed world, what we see because of that is a higher rate of allergies, allergic asthma, autoimmune diseases, including inflammatory bowel disease, Crohn's disease, irritable bowel syndrome, type 1 and type 2 diabetes, multiple sclerosis. And these again have dramatically increased in developed countries, but they remain at relatively low levels in undeveloped or developing countries and more agrarian parts of the world. Of course, in that area too, you still have more problem with fecal oral route diseases, intestinal diseases, and such. So there's quite a balance going on between microbes in the body, but they are critical to our normal function from the point of regulating our metabolism and regulating our immune systems. And when we alter our microbiota, that can alter our metabolism and that can alter our immune responses. Now remember, at the end of every SoftChalk lesson, you do have a self-quiz covering everything in that lesson. So again, you want to make sure you go through and do these self-quizzes to quiz yourself, see if you understand that material. And if you go through and you don't get most of them right, then you want to reread and relearn that material, look at the illustrations and such, and try it again. So that ends our first SoftChalk lesson in Unit 1, Lecture 1.